8, I believe. It used to be chapter 9. I'm still wedded to the old textbook. It's been, I don't know how many years now, but I'm still wedded to that old textbook.
Chapter 9.
Is it chapter 9?
For like the actual tissue.
So is it muscle tissue is 9? Okay, so then it used to be 10. The vocabulary is significant. And some of this vocabulary, I looked at an old diagram that I drew, and I messed up the vocabulary too. So, and it's not that it's that hard. It's that it's just a lot. Let me just get this my barely sufficient workspace organized here. So the only thing that we're going to do today is doing a drawing. That drawing will be probably it'll be multiple pages. So and I will post them later on today or tomorrow, depending on how it goes today. So Mondays I have busy days with my afternoon class. One of the pairs of terms that I always screw up are the differences between ligaments and tendons. So. So this is chapter 9, right? Okay. So in terms of the lab, in terms of the lecture, we're going to talk about the lecture. We're going to do this drawing, and then we're going to take a break, then we're going to finish this drawing. We're going to talk about the quiz, which is next Monday, what the quiz will be on. And it will be on a combination of today and tomorrow's lecture, and then last week's lecture, which is on joints and articulations. Okay? In terms of the terminology that we are going to start using, the two terms that I always screw up, and I don't know why. I saw that on a drawing that I did a couple years ago, I messed this up. I've only been teaching this for a couple of decades, since long before you were born. I know the definitions of these two, and I still screw it up. So let's just start with making sure that I know what the hell I'm talking about. What is the definition of a ligament? So muscles to muscles don't really happen often. There's two examples of that are tendinous structures, but muscle to muscle generally doesn't happen. So there are bone to bone connections. And so which one is a bone to bone connection? Ligaments are bone to bone. And then what are tendons?
Bone to muscle.
And bone to muscle. In terms of the vocabulary, so part of building the vocabulary, if you see prefixes, there's two prefixes in particular. There's a prefix called myo and sarco. Whenever you see those prefixes, you know that you should immediately say that's something to do with muscles. And then the suffix, mycium. So all three of these have to do with muscle. when we're talking about muscle, we have really three types of muscle that we can discuss, right? What are the three types of muscles?
Skeletal, smooth, and cardiac.
Skeletal, smooth, and cardiac. In terms of this lecture series, this lecture group, we're talking about muscle tissue in general, but what we're really focusing on is we're really focusing on skeletal muscle tissue. So we're really worried about skeletal muscle tissue, and when I get done with skeletal muscle tissue, just like in the tissue section when we got to nervous and muscle tissue, I skipped over nervous and muscle tissue in chapter 4. When I get to cardiac and smooth muscle, I'm going to skip over that because that's a topic for AMP2. Regardless of what we're talking about here, we're talking about muscles that only do one thing, and that is to contract. So when they contract, every other thing that happens in our body due to muscles is a result of that contraction. And it's important for you guys to recognize that muscles only do one thing, so that if we're talking about a muscle that pronates, then, and you are supinating your hand, and if you don't know what I'm talking about, there's a vocabulary term, right? So if you're talking about muscles that are strong pronators and you just supinated, that muscle that's a strong pronator was relaxing. So they have two conditions that we're worried about. There's relaxing or contracting. Later on this week, what we're going to find out is that our muscles aren't used continuously. We don't use a muscle in an all or nothing, right? So our muscles themselves, especially the skeletal muscle, is voluntary. And because our voluntary muscles, we're able to gauge how much effort is required. We can use some of the muscle and then allow other part of that muscle, that same muscle, to relax. And then when the energy for that first part of the muscle runs out, then the muscle that wasn't being used, again, in that same muscle, let's just say the biceps brachii, another part of that muscle will take up the load, and the first part will relax. So now, in terms of the characteristics of muscles, skeletal muscle is striated and voluntary. Smooth muscle is non-striated and involuntary. And cardiac muscle is striated and involuntary. So the second two are both involuntary. The first and the third are striated. Striated means there is an organization of proteins that is set up in a particular way, and that organization produces this striations or stripes in the muscle. When we start talking about the muscles themselves, and this is the skeletal muscle now. When we start talking about the control of a contraction, we're going down to the very, very microscopic level of the control of the contractions. We're going down to the level of a protein that is covering another protein. And so we're going to be particularly when we're talking a skeletal muscle here, there's 5 proteins that control contractions. And those 5 proteins there basically they control contractions. So in terms of the 5 proteins, a few of them you might know. But the idea here is that if the proteins are present in one place, then the muscle does not contract. If it's moved away from that place, then the muscle does contract. So it's either an off or on switch. So this room has a dimmer switch, and so I can increase or decrease the light. But this is not at all applicable at the level of what's called the sarcomere, at the level of the contractile unit. So in terms of the skeletal muscle itself, the 5 proteins there, you might know some of them. The most obvious ones are the first 3, myosin, actin, and titin. The next 2 are, I grouped them as a unit, but we're going to put them separately today. Oops. So this is where we're headed. We're trying to understand how these proteins themselves, myosin, actin, titin, troponin, and tropomyosin, how these muscles or these proteins are organized. Because this structure is voluntary, it's controlled, the contraction itself is controlled by a muscle, by a neuron, and it's controlled by
Bless you.
Thank you.
It's controlled by motor neurons, and in particular, when I talk about the motor neuron itself, I'm not going to draw the entire motor neuron. I'm going to be drawing the axon, the terminal axon, and then a neuromuscular junction. So without this neuromuscular junction, then the muscle doesn't contract. Without these proteins, this muscle doesn't contract. So you can't get a muscle contraction with all of this working, not working together. So this is where we're headed. We're headed down to the cellular and subcellular processes. But let's start from let's go back to where we started talking about. So when we're talking about the muscle itself and the bone, we're talking about the connection between bones and muscles here. Let me just see how much of a picture I have here. All right, I think I can do it. So what I'm going to do now is hopefully something that is familiar, and hopefully I practiced it last weekend on this thing, and I did a pretty good job, but let me see. Ms. Angel would not think this is anatomically correct, but it's close enough. You hopefully will think that this is a bone, and in particular it's meant to be the femur. There's a couple of processes that hang off of the femur, and those processes are the trochanters, right? So I want to just remind you of a couple of things. So the main shaft is the diaphysis. This is the proximal epiphysis. And the blue line that is on to the right of the diaphysis is the periosteum. The periosteum is very important in this because it is continuous with this next structure that I'm going to check again and make sure I don't screw up. And when I label that, that's the tendon. So the periosteum is continuous with the tendon. So the dense connective tissue that is produced by the fibroblasts and the fibrocytes that are how fibrocytes are producing those collagenous fibers. Those dense connective tissue there is continuous with the muscle. And so it's this connective tissue that wraps in many, many, many different layers. It wraps the muscle, okay? So what I'm in the process of drawing right now is the muscle itself.
It's like Dory.
What?
Dory.
So-
No, I can't draw that. I don't know where you see a door.
Dory. Let me see the bottom. Wait, never mind.
And there's a series of tubes that the muscle is consist of. So what I want you to think about here when we're drawing that is that imagine I take us all to KFC. I used to love Kentucky Fried Chicken, Popeye's chicken, right? So you take off the crispy epidermis, right? We know that we're integumentary system, right? We take that off, right? And then we're going to just say it's a drumstick, right? Now we're going to pull the drumstick, the meat off of the drumstick, which is what? Which is muscle tissue. It's skeletal muscle tissue. So we pull that off. Does the entire muscle come off? What are you looking at me for?
Right?
Oh,
Like-
I'm sorry. So this is, I'm not trying to be inappropriate, but this is true, right? This is just the way it is, right? I think it's a little bit grosser if I start talking about eating my biceps brachii, right? But if you start pulling off the drumstick, a little bit of the muscle is gonna come off and a little bit more of the muscle is gonna come off. And one of these is gonna come off and another of these is gonna come off. And then as you break those up, every once in a while you see this long fiber of muscles. And that's what we're trying to get to here, okay? So that first, there's an outer layer of tissue that we're gonna talk about in a second. This is the entire muscle. And then as you pull off remember now the integumentary system, as you pull the integumentary system off, you notice that there's some connection between the integumentary system or the hypodermis and the muscle itself. Well, that connective tissue now, because it's got cooked, is broken up. All of the layers, the tendon isn't broken up. The ligaments aren't broken up. And so some of those are real chewy, right? They're grizzle and stuff like that. You don't want to just sort of take those and spit them out, right? That's what I do. So that stuff you want to get rid of, but you want to get to the meat. So these things that are successfully smaller and smaller parts of the muscle are what I'm trying to draw here. So these individual ones, are labeled something. First of all, this is the entire muscle. And if you look at the top, right above this entire muscle, and I said there's the mycium right here, the covering of the entire muscle is called the epimycium. And then we have that entire muscle that is then broken up into smaller and smaller subunits. The first subunit that it's broken up into is called a fascicle. And that term fascicle is just a unit, is basically defined as being a unit of, in this case, a unit of skeletal muscle cells. The unit of skeletal muscle cells is covered by the perimycium. And embedded into this, into this muscle tissue are these things right here, these. And I'm gonna try to label this with blue. So this muscle has lots of fascicles. So there's a fascicle there, there's a fascicle there, and there's other fascicles. But what I have in the lower, with the red, the blue, and the green, is a compartment of both nerves or neurons. Well, no, at this point it's nerves, and it's motor nerves, arteries, and veins. So there's motor, that's motor. Motor nerves, arteries, and veins. And as we get successively smaller and smaller, those motor nerves, arteries, and veins get smaller and smaller, and they then have a different terminology. So you may hear me using the word arteriole and venule. That's a smaller artery or vein. And then ultimately you get down to the level of the capillary, and that level of the capillary is the smallest blood vessel in our body. So now, if you talk about, this is a single fascicle that I've been drawing to the right. And it is got more of these fibers. So this single fascicle so that one fascicle, which is covered by the paramycium, is a combination of muscle cells. Terms for muscle cells, there's a number of different terms that people have used over the years, and I'm not sure why it's so sort of, there's such a rich vocabulary of something as simple as a muscle cell. Do you guys have a question? A myocyte or a myofiber is at least two terms that mean muscle cell. And when I say muscle cell, I'm just talking about skeletal muscle cell. So now, one fascicle is a group of muscle cells that can either be myocytes or myofibers. So that extension that is going to the right is a single muscle fiber. And that single muscle fiber then is got individual fibrils, and those fibrils are look like they're darkened. There's a series of dark and light sections of it. Those individual myofibrils, I'm going to tell you what they are in a second here, is what we're trying to get to. So this is so that one myofiber itself is covered by more connective tissue, and that connective tissue is called, instead of the paramycium, at this point, it's the endo. In terms of the fibers themselves, the coverings, there's different layers of coverings here. So there's, from the deepest to most superficial, there's endomycium, paramycium, and epimycium. And then if they all merge to form the tendon, and then those merge seamlessly with the bone. It's a little bit like taking, if you think about a textbook, and if I take an individual page of a textbook, it's very easy for me to rip. If I take an individual endomycium, it's very easy for me to rip. But if I then go to the entire tendon, which is just a bunch of pages of endomycium, if I take the entire tendon, right, that's going to be very difficult for me to rip. and how difficult it is, even after you cook it under very, very intense heat over very many minutes, right? That tendon there doesn't degrade. So that's how strong it is. And in contrast, the endomysium and the paramysium are things that are going to be breaking up. There's another cell here.
Yes, sir.
Or another two cells, really. This is, the green part is a motor neuron. It's a motor neuron, and the part of that motor neuron is actually called a terminal axon. That terminal axon is covered by neuroglia, and these neuroglia cells are basically enhancing the function of the motor neuron or the terminal axon, which is to send action potentials to the muscle. And the term is action potentials. I'm going to make this green part a little bit bigger. I just remembered something. So the idea here is and I put arrows down in blue that go from the upper left. The arrow there's this arrow right here is sending the signal down the terminal axon to the end of that terminal axon. And at this point right here, that thing right here is called a neuromuscular junction. So that neuromuscular junction is when the neuron, or the terminal axon in particular, almost touches the myofiber or the myocyte. It doesn't touch it, but it gets really close. In terms of the end of the terminal axon, we have these organelles that are characteristic and of a typical cell. There are these organelles that have stuff inside of them. In at the neuromuscular junction here, let me just write this up here. So we're talking about, we have mitochondria. And what are they going to do? They're going to produce ATP for, we're going to learn more, or remind yourselves of the sodium-potassium pump. They're going to be basically setting up the sodium-potassium pump so that action potential can get set down there. And that is this drawing right here. And then these little circles here that are right next to the mitochondria are called synaptic vesicles. And those synaptic vesicles have this thing called ACH. And there's a bunch of these things, but one of the most important ones is called ACH. For neuromuscular junctions of skeletal muscle tissue, ACH is very, very important. And this is called acetylcholine. Acetylcholine, or ACH, is also known as a neurotransmitter. And all that we're going to be doing here is talking about how we have the stuff that's found in the motor neuron, in this terminal axon. It leaves that cell, it crosses this little space between the green and the black, and then it goes into the black. So that in terms of cellular terminology, we're talking about exocytosis, simple diffusion, and then receptor-mediated endocytosis entering the cell. So that terminology is something I expect that you remember from chapter 1 or chapter 3. And it's going to be coming, we'll talk more about that in a little bit. So first of all, are there any questions right now? Now, let me go back to this part right here. This, I just wanted to tell you that so this group of muscle cells also has blood flow, but that blood flow is much, much smaller. And there's lots of blood flow going there. And if you've ever worked out and had pain from that you had, pain after exercising, my wife and I went for a bike ride yesterday, and we were both like, oh man, my back hurts a little bit. We're a little bit sore here. That soreness is a problem with some of the muscle cells. The muscle cells themselves have these these stripes, these dark and light areas. And again, that's due to organization of the individual myofibers. This one myofiber has many, many, many myofibrils. And a myofibril, by definition, is a stack of sarcomeres. If I had more markers, I could connect the markers together in successive markers, and I can make a very, very long pile of markers, right? Each marker, in this case, would be an individual sarcomere, and then the connection between the markers is the definition of the end of the sarcomere. So if this is, the sarcomere itself, we're gonna have to talk about what's happening, how is the sarcomere organized, how is it, what are the proteins and what do they look like, what's happening here, and when it gets from here to here, then what happens to individual myofibrils. So when you're talking about a muscle contraction here, we're talking at the level of a myofibril, one myofibril, and one part of a myofibril, okay? So we're getting down to the some very small layers here. Let me see something. Now, so this one myofibril is a stack of sarcomeres, and the sarcomere, and I'm going to talk about what the sarcomeres are right up here. So now, when we talk about sarcomeres here, they have two conditions. The sarcomere is either fully contracted or fully relaxed. There is no in between. So then the question is, and I once, when I was a graduate student many years ago, remember the professor in anatomy would say, And he would always get a laugh by this because he was a very, very light-skinned bald guy that had alopecia. He had no hair, had nothing. He was very unique. Anyway, what he would do is say, why can't I stop hitting my face? Why is it that I want to touch my face, but I keep, and he would do this, and he'd start smacking himself in the face, and he would turn red, and so he had people, like 100 people laughing at him. And that idea here, an individual sarcomere contracts either completely or not at all, but your muscle doesn't do this, doesn't contract completely or not at all, right? Your muscle can stop here. Your muscle can stop here. It can go back, right? Sarcomeres don't have that option. Right? If the sarcomere is going to contract, it goes all the way, and then it comes back. So how does that happen? Another question is, if the sarcomere is contracting and then gets damaged, how is that repaired? And then the final question is that if your muscles change in size and shape, what happens to the muscle cells? And when I'm talking about muscle cells, what am I saying? I'm talking about the myofibers or the myocytes. If you go and you start working out and you get bigger muscles, do you get more muscle cells or do you get bigger muscle cells? And we'll talk about that in a second. But let's get to the level of the sarcomere. And so it is a functional unit of a skeletal muscle cell. And when we're talking about sarcomeres here, we're going to talk about a couple of things here. There is multiple sarcomeres that I'm about to draw here, but in terms of the terms, I'm going to talk about them being AMHIZ. Right, so AMHIZ, and we're going to talk about the sarcomeres, and there are two conditions. There's two conditions. There's either contracted or relaxed. So the first thing we're going to do is draw relaxed sarcomeres. And I'm going to draw parts of 3 sarcomeres. I'm going to draw one entire sarcomere with a little bit of one on the left and a little bit of one on the right. In my drawing right here, we have a little bit of this between these vertical black lines. That's the entire extent of a single sarcomere. And a little bit to the right is another sarcomere, a little bit to the left is a third sarcomere. In this drawing right here, we have that center line. And then we have this thick red stuff. Oh, wait a minute. And my drawing is not perfect by any stretch. It's reasonably accurate, but what the drawing here is meant so that the green lines are meant to be lined up vertically. The end of the red lines are meant to be lined up vertically so that everything is there's this certain overlap right here. Yeah. In terms of the muscles themselves, there's the thick one. The thick red one is myosin. There's actin, and those actin and myosin are the ones that are most famous or most well known. Those are the contractile proteins that we're talking about here. And really, when we're talking about the contractile proteins here, we're really not talking about 2 proteins that are contractile, we're talking about 1. The other one goes along for the ride. In this drawing right here, I've gotten this green squiggly line. And the green squiggly line, just so we're clear here, I'm not gonna continue on with this drawing, but we have a part of a sarcomere to the left and a part of the sarcomere to the right of this. So I just wanted to be explicit here, or hopefully explicit, and tell you that the sarcomere, so there's 3 sarcomeres again, parts of 3 sarcomeres. This squiggly line right here is called titin. So right there, we have 3 of the 5 proteins that I mentioned. I mentioned these 3, actin, myosin, and titin, and then we'll talk about troponin and tropomyosin. Now I want to label this, and there's 5 parts to this. The M is the middle of the sarcomere, and the end of the alphabet is Z, and so that's the end of the sarcomere. So this is called a Z disc, and that is the Z disc. And that's the M line, the middle of the sarcomere. So the sarcomere has 3 more areas. So for each sarcomere, you have the I band on one side and then the I band on the other. And then the last 2 here, the A and the H. I never can draw the A and the H very well. So we're going to try to define what that is in a second. So in terms of the A band here, the A band is called the zone of overlap, the overlap between actin and myosin. So I was trying to include the entire length of myosin. The entire myosin molecule. The I band is the area where there's green here. It's a combination of actin and then the very, very thin protein called titin here. So this is the light area here. This is and then the H zone is just the area of myosin only. Now, when I have a question for you to identify what's this area, what's that area, what's this protein, what's that protein, I'm only going to ask you for a relaxed sarcomere, okay? Because what I'm going to do next, in the next drawing, is we're going to take everything here, and I'm going to draw a new one down here. And what I'm going to say is the next one is we only have 2 conditions for sarcomeres. There's relaxed and there's contracted. Okay, so it's important to recognize that this one will be fully labeled, and I'll ask you lots and lots of questions about relaxed sarcomeres. And then in terms of drawings and then photomicrographs or histological sections, you need to be able to recognize what a contracted sarcomere looks like versus a relaxed sarcomere. And the idea here is that for a contracted sarcomere, there is no area of light and dark. And for a relaxed sarcomere, there is. So this area right here is going to be dark. This area right here is going to be light. Why is it going to be light? Because it really only has this one narrow protein, so it's one little very thin protein. So this little thin protein, actin, chitin really isn't very visible. The thin protein shows up as light. Myosin, the thick protein, shows up very dark. Okay? So let me see if I can do that. Well, not if I can do it. Let me see how it works. All right. So now for this part of the drawing, what I'm going to try and do is I'm going to try and copy the M line right down the middle, right down there. Then I'm going to try to replicate the length of the myosin molecules. And then things will change. So the first part of this is going to be the M line. And then the myosin molecules, 3 of them. We're not labeling this because this is going to be a contracted sarcomere. So this will be a contracted sarcomere. Now, in terms of the actin is going to be the same thing right here. You really can't tell the difference between the actin in the top drawing and the actin in the bottom drawing. The one thing you hopefully can see by my drawing is that there is more overlap between the actin and the myosin. So that zone of overlap has increased. There is still the titin that is present. So in other words, the entire structure, physical structure of this is the same. Okay? The one difference between a relaxed sarcomere and a contracted sarcomere is that one part of the sarcomere itself, all it does is it goes this like this. Okay, so the entire sarcomere just gets this, it contracts fully. And so what does that look like? Let's see here. These vertical black lines are the Z discs. And that looks like a 2. So the 2 Z discs. Now, the definition of a contraction then is just the movement of the Z lines or the Z discs closer together. That's all a sarcomere. That's what happens in every muscle. That's what happens in every myofibril, and that's the definition of a contracted sarcomere. Okay. So now, when I show you pictures tomorrow, they're going to be what type of sarcomere is this? What type of sarcomere is that? Right? So they're generally going to be labeled, and I'm going to show you pictures of it, relaxed and contracted. At the end of the day, these are 3 of the 5 proteins that we're worried about. Now, let me see here. So what I want to do now is talk about the other 2 proteins, and we're going to talk about the protein there as a complex here. There is troponin and tropomyosin. I think I have enough room over here on the right, and then we're going to take a break, and I'm going to try and copy this and put it into another file. So the idea here is that the troponin and tropomyosin portion of the molecules, those proteins there, they sit on actin. Their job is to sit on actin, and when they're on one part of the actin site of the actin molecule, there's no contraction, and when they move, there is a contraction. So they're the light switch of a muscle contraction. So now the troponin. So the last two proteins are called troponin, tropomyosin, and I'm talking about them in a pair. They reside on top of actin. They reside on the actin active site, and all they do is when they're on the site, then it's relaxed, and when they move, and when they're moved, and they're moved by an ion here, a cation. When calcium comes to reside there, the calcium moves the actin off. Moves the troponin tropomyosin off the active site of actin, and when it's moved off that active site of actin and ATP is present, then you have a muscle contraction. Okay, so. Are there any questions about this? So what I want to do now is I want to take, let's see here, it's almost 9:30. I see a couple of you are sleeping right now, so I want you to, Jelly? Hey, Jelly, can you sit up? Or leave. I want you guys to get up and walk around. I'm going to save this file. I'm not going to upload it yet. I'm going to check it first, okay? So it's 9:30, let's come back at 9:40.